Welcome back to another learning series with Mr. Knight. Do you remember that photocopying incident in the office? Well, today the office is much more busy. We are having two sets of copying taking place, and papers are just all over the place. Now, in your bodies, similarly, copying is taking place, and there are two sets of division. So today we're going to look at the number of chromosomes involved in meiosis. If you haven't seen the lesson on the numbers of chromosomes involved in mitosis, now is a good chance to look at it. In fact, it will be at the end of this video, so you could click and watch. Now, to remind you, is that inside of our body we have chromatids. Chromatids are only a part of a chromosomes. A chromatid, when it joins with a centromere, forms a chromosome, and there are two alternative ways. On the left, we have one chromatid, one centromere, which makes one chromosome. On the left, you have two chromatids, one centromere, which still makes one chromosome. Now, the idea in this, the number of centromere represent the number of chromosome. So on either side, there is only one chromosome because there is only one centromere. Now, let's debrief again before we copy. Think about a chromatid as a sheet of paper with information. What information is there? Genetic information. Think about a centromere as a binder that holds papers together, as the centromere holds chromatids together. Now let's put this into perspective. Let's say you have a stock of paper to be copied. What represents a stock is the binder. So binder represents the number of stock. So right here you have one stock of paper, but only one sheet of paper. If you're going to copy this, will you copy the binder? Absolutely not. So therefore you will only copy the paper with the information. You put the paper in the printer, you result in two copies. If you choose to put the binder on both papers, you only have one stock, but two sheets of paper. Let's look at the alternative method. The same one sheet of paper, one stock, you do not copy the binder. You photocopy, you realize now you want one stock, you put one binder. So you have one binder, one stock with two sheets of paper. If you separate the sheets of paper and want to maintain them being organized, then you put separate paper clips or separate binders on each paper. So hence you'll have two stocks for two binders and two separate sheets of paper. So two stocks, two sheets of paper. Now let's apply this to chromosomes. You put one chromatids in. Remember, you're not copying the centromere, only the information part. When you copy that and you choose to keep them together because of one centromere, you have one chromosome, but with two chromatids. Alternatively, if you decide after copying to separate the chromatids, you'll have two separate chromatids with their own centromere, which means two separate chromosomes, but a total of two chromatids. Now for meiosis, there's a little trick. Something that I want you to remember is that in meiosis, there are two sets of metaphase, and in the metaphase is where things are interested. Now, the first metaphase, call this one cell. That's one cell. The chromosomes, notice I use the word chromosomes. The chromosomes align side by side or parallel to each other. However, in the second metaphase, because after the first division you have two separate cells, and each cell, the chromosomes will be aligned in single file along the metaphase plate. And then by the end of that second division, they will separate it into four separate cells with half of that number of chromosomes that were in the metaphase. All right, so now we understand that. Let's go a little bit further in looking at some numbers. Now, do not get frustrated with these numbers. These numbers will be explained and made easily. Remember now, there are two sets of division. So let us go back to our story. Let's tell you a story about these numbers first for ease of understanding. Now, this is a story about I Preserve Myself 2. So if you haven't watched I Preserve Myself 1, check the mitosis lesson. So I Preserve Myself 2. So remember again, we have two sets of division. First, you have one interphase. And make sure I mention that before I go into the story. You have one interphase, only one. And then you have PMAT1. So that's why you have P1, M1, A1, T1. So PMAT1 and then a cytokinesis. So let's go into the story. Once upon a time, there were two sisters. One called Chromatid, the other one called Chromosome. They were living lovingly as two sisters. However, something interferes, and causes chromatid to double in size. Chromosomes preserve herself and maintain that love. It maintained love right through. However, coach still stepped in and cut them down into size. Even after this cutting down to size, chromosomes still maintain its love and preserve herself. However, at the second part of attitude stage, chromatid decide to have an attitude and become antisocial. Then chromosome just level up. And while chromosome level up, the coach realize and come and cut them down into half their size. That's how they start to live lovingly ever after and start to reproduce. Get it? Sexual reproduction. Now let's look at some numbers. Now, this is long, yes, but because there are two sets of division. So let's take this stage by stage. First division, meiosis 1. Second division, meiosis 2. Meiosis 2 is just like mitosis. So again, check that mitosis video. First, let's start with original cell in human beings. 46 chromosomes, 46 chromatids. So 46 chromosomes, 46 chromatid. Remember, the only thing that is being copied, and is only copied once, it is the chromatids. This number, when it's being copied, is double. So it maintains this number all the way through until T. Now, interestingly, remember, for this, because of chromosome, remember what happened in mitosis? She didn't want to level up early. She didn't want to cause any problems. So she preserved herself. Chromosome is preserving herself and maintain this number right through to T. However, the coach, a little bit suspicious and cut them down to size. Just in case they want to misbehave, the coach came and cut them down to size. Now they go into a second phase. And the chromatid will maintain this number because there's no more replication taking place. So chromatid maintain the number straight through. However, there was an attitude at the anaphase, because this is just like mitosis, and the chromosome level up, yes, level up, right? And so the leveling up taking place because of that antisocial or attitude. And then the coach came in and cut them down to half their size. So notice both of them cut down to half their size. So if you notice, at the end of the second division, compared to the original cell, each original cell will have 46 chromosome, 46 chromatid. However, by the end of meiosis 2, each cell will have 23 chromosomes, 23 chromatids. And that's how they stay in half, and because they want to grow back in size, they want to reproduce. So want to reproduce. Reproduction will make you double up, right? Now let's look at an example. An example here will be using an original cell with 2 chromosomes, 2 chromatids. Remember, interphase will double up what? The chromatids. So the chromatid number maintain right through. So you don't need to worry about the chromatid number, they maintain right through. So if you count from the I to the T, there are 4 chromatids each. However, remember now, the chromosome as a memory or an history from mitosis, and remember, do not act up. So chromosome maintainer cool, and keep this number 2, 2, 2, 2, 2 centromere, 2 chromosome right through. However, the code's still suspicious and cut them in half their size. Now go to the second phase. Is that coming from the first phase? Since they're cut down to half their size, 4 and 2. So the 4 chromatids cut down to 2, and the 1 and the 2 chromosome cut down to 1. So let's go back to that. 2. So in meiosis 1, we have the 2 chromosomes, 2 chromatids, 1 chromosome. All right? And so what is happening here now? They maintain this number straight through. So the chromatid number maintain straight through. However, now the chromosome now going to be a different. So notice the difference right here now. From the P and the M, that number was maintained. Where we reach A with attitude start, just as mitosis. Then the chromosome level up with chromatid. And then the coach came now and do what? Cut them into half. And so if you notice, you start from 2 in each cell, 2 chromosome and 2 chromatid in each cell. That's original cell. So let's go back to the original cell. The original cell starts with 2 chromosomes, 2 chromatid. At the end of meiosis 2, you should have 1 in each cell. 1 chromosome, 1 chromatid in each cell. So you can pause, you can go back and review and look at these numbers, look at the examples until you understand. I hope this helped. And so at the end of the lesson, hope you have fun with these numbers in chromosome and chromatid. The mitosis button is right here. Click on it, you will see it. So see you in the next lesson, but do not forget to subscribe.